Welcome to this explainer. All right, we're jumping right in today. We are stepping way outside our normal, everyday scale and zooming straight into the microscopic world of plastic degradation. Have you ever wondered how scientists actually know for sure if a bacteria is eating away at our waste? Like, how do they prove it? Well, we're going to look really closely at a recent scientific study to see exactly how researchers have completely upgraded their forensic toolkit to measure how microbes consume bioplastics.
It brings up a really fundamental question. If a bioplastic falls in the soil, does it actually break down? You know, as consumers, we kind of just assume that compostable plastics easily degrade in nature. We see the word bio and we think, oh, it'll just vanish like a fallen leaf. But the reality of how and, more importantly, when these materials break down is a much more complex puzzle. It requires very, very specific biological and environmental conditions.
Chapter 1. The Stubborn Suspect.
So our prime suspect here is PLA. It's a hugely popular bioplastic. You'll find it in everything from disposable cups to agricultural films, but the temperature required to break it down is wild. At 58 degrees Celsius in an industrial compost facility, no problem, it degrades perfectly. But in normal, cooler soil, somewhere around 20 to 28 degrees, it's a totally different story. Its tightly packed polymer chains act literally like an unbreakable chain-link fence to the bacteria that are trying to eat it.
Chapter 2. The Flawed Clues.
To figure out if microbes could actually break down these stubborn plastics, scientists used to rely on a rather blurry visual test known as the classic clear zone method. Picture a cloudy gel packed full of plastic, right? And in the center, there's a colony of bacteria. If a clear halo forms around that center, that tells us the bacteria is eating the plastic. But here's the catch. It's a flawed, all-or-nothing clue. Sure, it confirms degradation is happening, but it absolutely cannot tell us exactly how much plastic was consumed.
Chapter 3. Upgrading the investigation.
To truly solve this puzzle, scientists had to upgrade to precise chemical math. But the very first hurdle they faced was time, because instead of waiting years and years for those stubborn PLA chains to naturally degrade, researchers cleverly modeled aged plastic using shorter pre-cut versions. These are called oligomers, or OLA for short. Think of these as pre-chewed bite-sized snacks for the bacteria. It dramatically speeds up the timeline, but still gives us highly accurate data.
But to track the exact chemical trail of these little snacks, the scientists had to meticulously map their starting point. The material was broken down exactly like this: 81% insoluble OLA, 8% soluble OLA, and 11% free lactic acid. Locking in those starting numbers is absolutely essential. It's the baseline for the forensic math they're about to perform.
Next up, they had to introduce this plastic feast to the bacteria safely, meaning absolutely no toxic solvents. The result is a cloudy plate completely filled with these tiny, water-insoluble microdroplets of olea. They're perfectly suspended in the gel and just sitting there, ready for the bacteria to attack.
Chapter 4. Following the Chemical Trail.
Okay, this right here is the real aha moment. This is where scientists transformed a physical petri dish into a math equation. As the bacteria grow, they secrete specific enzymes that act exactly like molecular scissors. They snip those larger OLA chains down into single lactic acid monomers, which they then eat up. And because scientists know the exact chemical signature of lactic acid, they can track its every single move.
To measure this whole vanishing act, the upgraded method uses a brilliant three-step process of elimination. Step 1, you chop the agar gel into manageable pieces. Step 2, you use water to extract and measure any free-floating lactic acid. And Step 3, you apply a strong base, specifically sodium hydroxide, to chemically force any leftover plastic oligomers to break down into measurable lactic acid.
It's a really elegant three-path setup. By mapping out exactly what washed out with water versus what was chemically broken down by the sodium hydroxide, researchers can just subtract those chemical leftovers from our original baseline. This calculation reveals, with just incredible precision, the exact percentage of the plastic that the bacteria actually ate.
Chapter 5. The 30-Day Verdict.
So what actually happens when we unleash the bacteria using this brand new forensic method? Well, that initial 11% of free lactic acid we established earlier, it drops to near zero, almost instantly, basically by day 1. The microbes are actively and hungrily devouring the absolute easiest food source straight out of the gate.
But the long game is what really matters here.
We're looking at the biodegradation kinetics, the rate of breakdown over a full 30-day period at a nice, optimal 37 degrees Celsius. And the data proves that whether the bacteria were introduced as dormant, sleeping spores, or as fully active cells, both populations had massive, sustained success breaking down those insoluble plastic droplets over the entire month.
And here is the huge payoff of all this careful chemistry, the final number. An incredibly impressive 70% biodegradation rate achieved in just one single month. I mean, that is a massive leap from just guessing by squinting at a blurry halo. It finally gives the scientific world a precise, high-confidence measurement of microbial efficiency.
What this explainer really proves is that by rethinking our lab tests and getting creative, we can completely move away from merely guessing if a plastic degrades. We are now capable of knowing exactly how efficiently nature can clean it up. And that is a real game changer that opens the door for far more accurate environmental testing. With this highly precise forensic toolkit now totally proven to work, it leaves us with one massive question. What other incredible plastic-eating supermicrobes are hiding right beneath our feet, just waiting to be discovered and properly measured? The microscopic hunt is truly just getting started. Thanks so much for joining me on this explainer, and keep questioning the science around you.